Protozoa can be further classified in the attributes protozoa the different classes of the protozoa. Similarly, we can classify elements, which are the multicellular organisms, into cystodes, nematodes, and eukaryotes. So today we are going to discuss the Leishmania and the toxoplasma. The Leishmania belongs to the class flagellates, while the toxoplasma belongs to the class sporozoa. Leishmania, Leishmaniasis is caused by Leishmania is a parasite. These are hemoflagellates. These are related to the blood. That's why we call it as hemo. We have the flagella, so that's why we call it as the hemoflagellates. So leishmaniasis is caused by the leishmania. Leishmaniasis is a disease basically. It's a zoonotic disease. Zoonotic disease, the vector is involved in the transmission of the disease. Leishmania is a parasite which is causing the leishmaniasis, and the transmission of the disease is via vector, and the vector is sandfly, female sandfly. This transmitted in the mammalian host by a female sandfly. There are different species of the Leishmania. Leishmania donovani, Leishmania tropica, Leishmania mexicana, Leishmania brasiliensis. Three important species are Leishmania donovani, which is causing the visceral leishmaniasis. It involves endothelial tissue, livers, spleen, and bone. Leishmania tropica and Leishmania mexicana, it is causing the cutaneous leishmaniasis involving epithelial cells, the skin, and the site of sandfly. Leishmania brasiliensis, which is causing the mucocutaneous leishmaniasis involving mucous membrane of the mouth and food spread from a nearby cutaneous lesion. The important properties is vector is involved in the transmission of the disease, and the vector is the female sandfly. Because only they take blood mean for egg maturation. They take the blood, ठीक है, जो भी infected person है उससे ये blood लेंगे, then within the sandfly there is maturation of the egg. There is the male and female gamete union, and that leads to the egg maturation. Sandfly ingest macrophages containing amestigotes. I will explain you all these. So this is the life cycle of leishmaniasis. The promastigotes. Promastigote is the form of the parasite, which is the flagellated form. Now they are converted into the aflagellated form once they enter into the body and they are taken up by the macrophages. There is no flagella in this, and they are converted into the aflagellate form. This is known as the amastigote. The promastigote stage inside the cell wall. Here you can see the sandfly. This vector is carrying the flagellated leishmania. Promastigote stage of the leishmania. This is present within this sandfly. Now this is the life cycle of leishmania. Now here you can see the sandfly take a blood meat, okay, inject promastigote stage into the skin. This sandfly is infected with the leishmania. This leishmania is parasite or protozoa is present in the form of the promastigote. It means they have the flagella. Right now they are introduced into the body. These promastigotes, they are phagocytosed by the macrophages. Within the macrophages, these promastigotes, they are transformed into the aflagellated stage. This is known as amastigotes, and now they multiply within the macrophages. After the multiplication, time will come when they burst out and they come into the macrophages will burst out and they come into the circulation. Now another sandfly can bite infected human and take a blood meal which is containing the amastigotes. Okay? Now these amastigotes, they are taken up by the sandfly. These mastigotes are transformed into the promastigote stage to the midgut of the sandfly. Now divide in midgut and migrate to the cycle. Okay. Now the sandfly is again ready to bite the another healthy individual. In this way, the whole cycle repeats and is causing the transmission of this leishmania or foot parasite. The visceral disease caused by the leishmaniasis, which is leishmania, this is known as the cancer. The most severe form of disease, the disease typically starts with irregular bouts of fever, chills, and general edema. Since leishmaniasis is primarily a disease of the reticuloendothelial system, replacement of infected cells induce hyperplasia and consequent enlargement of the visceral organ associated with the system, example spleen and liver. Like these, in this disease, we can see the splenomegaly, the lymphatosplenomegaly, along with anemia and fever. Post-Calazar dermal leishmanoid can be seen after 2 years of the specific skin. Some people recover spontaneously, some people who were treated later develop post-Calazar dermal leishmanoid. Lab diagnosis. The amestigote stage of the parasite can be seen in bone marrow, spleen, lymph nodes, biopsy. Serological, we can perform certain serological tests and skin test is positive in recovered patients. This is the picture showing you the bone marrow and the amestigote stage of leishmania. Leishmania tropica, Leishmania mexicana, then the sandfly is the vector and the forest rodent is the main reservoir. Leishmania tropica and Leishmania mexicana causes cutaneous lesions and the granulomatous response occurs and then necrotic ulcers form at the site, at the bite site. Here you can see this picture. At the bite site, this type of lesion can be seen and produce skin ulcers on the exposed part of the body such as face, legs, and disease can produce a large number of lesions. A mucocutaneous leishmaniasis causes the Leishmania basilensis, including the papilla and the bite site, but then metastatic lesion forms at the mucocutaneous junction. Their diagnosis amestigotes and leishmania taken from the skin. Another important parasite we are discussing, Toxoplasma gondii, causing the toxoplasmosis, including congenital toxoplasmosis. All parasite states are infectious. Less good are the pregnant women and the meat handlers. Definite host is the domestic cat, and the intermediate host are humans and other mammals. Mode of transmission, infection begins with ingestion of cyst in undercooked meat or accidental contact with cyst in diagnosis. It means food or placenta transmission. It means the contaminated meat or food can be a source of the transmission of the disease, or the contact with the cat feces, which is infected with this or carrying this toxoplasma. And the transplacental transmission from mother to fetus is also there. So here you can see this is the definite host. Definite host is the one in which the sexual reproduction takes place, right? So if this cat is infected with toxoplasma, suppose this cat is infected with the toxoplasma and it's carrying this cyst, these parasites are present in the form of the cyst. They shed into the feces and they are sporulated oocytes. Right? Now they come into the in the environment and they contaminate the food and water, or they may directly affect the pregnant women by the contact of the pregnant women with the cat feces or any other accidental. Now, first we will discuss the contamination. After the contamination in water, this may be taken up by the intermediate host, other mammals. Now, this meat is eaten by or taken by the definite host of the mammals. So, within the tissue, they are present in the form of the breviozoids. Now, this tissue or the meat is taken up by the carnivores, and cat will ascertain, humans will ascertain, and in this way there is a transmission of the disease via the contaminated water. Now, after exposure of these cyst or the toxoplasma that is when the may be present in the feces of the cat of the pregnant women. After exposure of the pregnant women with this contaminated feces, they may transmit the disease. They may get the disease, pregnant women. Now, we once they entered into the body, they are present in the form of the techizoids. So these techizoids can cross the placenta and can affect the fetus. So techizoids can be transmitted through the placenta. But this form radiozoids, they are present in the tissue. They cannot be transmitted via placenta, or they cannot cross the placenta and cannot infect the fetus. Now this is the definite cat. Okay, this is again the similar picture. This is the cat which is carrying this parasite. Now they are taken up, or they contaminate the environment and the food and water they are taken up by the other mammals. They are present in fish. Now they also infect these mammals. They are present in the tissue in the form of the radiozoids, and they are taken up by the definite host or the other human beings. So there is a transmission of the disease. Or they may be the contaminated feces, maybe especially may come in contact with the contaminated feces and can cause the disease. But remember this phase is only occur in the pregnant women. If the infection occurs before the pregnancy, then there is no chance of the transmission of the disease. Only when the pregnant woman can get infection, then there is a chance of transmission of disease. Both oocysts and tissue cysts transform into techizoid shortly after the injection, because these oocysts and the tissue cysts, they has to be transformed into the techizoids after the injection. And these techizoids localize in neural areas of tissue, develop tissue cysts, radiozoids. Pregnant women become infected. Techizoids can infect the fetus via the bloodstream. Then mediated immunity play an important role. It may limit the disease, but if anything which is affecting the cell-mediated immunity, there may be chances of infection. Congenital infection of fetus occur only when the mother is infected during the pregnancy, before the pregnancy, and after the pregnancy. If the mother is affected or the woman is affected, then there is no chances of congenital infection. Patient may be asymptomatic. Patient may present with negative hemoglobin antibody test. Congenital infection, the leading cause of blindness in children, and results in abortion, stillbirth, encephalitis, chorioretinitis, hepatosplenectomy, fever, jaundice, and intracranial calcification. Lab diagnosis: microscopic examination of Genzel stain preparation shows decent shaped protozoids, and we can perform this test. This is all about the bladder tissue.